Thank you very much, Chuka, and thanks for the invite to come and talk today. Chuka mentions that we've known each other for 29 years, which makes me feel really old. I was giving a keynote talk at one of the European meetings recently, and I was introduced as the father figure of European liver immunology, which I was quite flattered about, but also a little bit insulted. I don't really feel that I'm a father figure yet, but never mind. So my two real interests and passions are clinically liver disease and, as Chuka mentioned, immunology. And just before I start the talk, one of the things I did more than 25 years ago, in fact, when I was a senior registrar in gastroenterology in the West Midlands, against advice from all of my peers, was to resign from that rotation and go to the National Institutes of Health to do a postdoc in a basic immunology lab with a fabulous guy called Steve Shaw. And for any of you younger members in the audience who are looking at developing a research career, I can't stress too much the importance of going and working with outstanding people. So when I went to work with Steve, it was just at the beginning of the revolution around chemokines and cell trafficking, and we were able to demonstrate how chemokines are presented on the endothelium of blood vessels to trigger recruitment of cells into tissues. And that's an experience and a training that has lasted with me for the rest of my career. And it underpins much of what I'm going to talk about today. So that's just a piece of advice from an aging clinical academic. So what I'm going to talk about today are extraintestinal manifestations of inflammatory bowel disease, particularly from my context, the liver manifestations, but we'll talk a little bit about some of the others as well. So if we look at how IBD can affect systemic systems, we all know that you can see eye disease, skin disease, arthropathies, and indeed lung disease in IBD, as well as the liver diseases, which we'll talk about, PSC and autoimmune hepatitis. And some of these extraintestinal manifestations are relatively easy to explain. So they correlate very well with episodes of inflammation in the colon in particular. And you can imagine possibly that this is due to activated immune cells becoming trapped in tissues and driving local inflammation and damage. And when you treat the underlying inflammatory bowel disease, these complications tend to resolve in parallel. And this is true also for some of the arthropathies, particularly the so-called type 1 porcine articular arthropathies. When you treat the IBD, the joint disease resolves and gets better. But some of the other complications, particularly the type 2 arthropathies and ankylosing spondylitis, and in the context of what I really want to focus on today, the liver disease, primary sclerosing cholangitis, and autoimmune hepatitis, these complications run a course that's independent of bowel inflammation, and that, I think, is a really interesting concept, which we'll come back to. So if we think of the main liver complications of IBD, some patients develop autoimmune hepatitis, but the most important complication and one of the most enigmatic diseases in hepatology is primary sclerosing cholangitis. So this affects between 3 to 10% of patients with IBD. And if you look the other way around, in Western societies, at least 80% of patients will have colitis, though often that will be microscopic colitis, which is asymptomatic. So you have to look hard to find the colitis. But the association is really strong and dramatic. So PSC is a terrible disease. It has a 15% lifetime risk of cholangiocarcinoma, an increased risk of colorectal carcinoma. And in a recent large study, UK and Scandinavian study, about 36% of patients will either die or come to liver transplantation as a consequence of their disease. And at the moment, we have no effective treatment. So this is a really dispiriting disease to treat, apart from transplantation. So what about the inflammatory bowel disease, PSC? Well, this colitis has some distinct features, and it is predominantly colitis. Even the patients with PSC who have Crohn's disease tend to have Crohn's colitis, not small bowel Crohn's disease. It's a predominantly right-sided colitis with pancolonic inflammation. The inflammation is often mild, as I've already mentioned. And a classic feature is a so-called backwash ileitis. So you see inflammation of the ileum as well as the colon. And what's fascinating is that ulcerative colitis can develop for the first time in patients who've been transplanted years before for PSC, and PSC can develop for the first time in patients who've had colectomies for IBD. That's emphasizing this dissociation in time between the two sites of disease. So one of the things that I've been interested in for many years now was trying to understand why there is this association and how can this association be independent of active disease within the colon. And we thought that maybe this could be because mucosal T cells activated in the gut become long-lasting memory cells. They recirculate for many years in the body, and then at a later date they can be activated and triggered by something in the liver, leading to the liver disease. But that original T cell activation is occurring with episodes of inflammation within the gut. So this is the hypothesis that the rest of the talk will be based on. So there's good evidence that this is not a silly model to come up with. And this is a lovely study quite a few years back now from Sue Bliss at Cornell, using a mouse model of Trichinella spiralis. So this is a parasitic worm infection. You infect the mouse by giving the worm orally. It goes into the intestine where the larvae are generated. It generates the CD4 T cell response in the gut, so you get a gut inflammation. The larvae then go via the portal vein through the liver, and in a normal mouse, there's no inflammation in the liver whatsoever. They go then through the liver into the circulation and cause systemic problems in muscles in the heart. But if you do this in an IL-10 deficient mouse, that's a mouse that is unable to regulate inflammation properly because it can't make IL-10, then exactly the same protocol results in a severe hepatitis, severe T-cell mediated hepatitis. And if you then take those IL-10 deficient mice and instead of giving them the bug orally, you actually inject it directly into the portal vein, so you bypass the gut, these mice are fine. So this demonstrates that under the right circumstances, T cells activated in the gut can cause liver disease. So is this tool relevant to what we're going to talk about with IBD and PSC? Well, again, there are animal models in which you disrupt immune regulation, particularly IL-2 pathways that are involved in the generation of regulatory T cells. And in these genetically manipulated mice, one of the characteristics is that they get inflammation in the gut and they get an inflammation in the liver, and that inflammation in the liver tends to be a cholangiopathy. And this isn't just relevant to mice, because the recent GWAS studies in patients with PSC suggest an incidence of defects in exactly the same pathways in humans with PSC as we see in the mice, particularly IL-2RA. So there's evidence that genetic defects around immune regulation can, under the right circumstances, lead to inflammation in the gut and in the liver. So how does this relate to these memory T cells I was talking about earlier, and to the idea that T cells activated in the gut might be aberrantly recruited into the liver? So we know now from many studies over 20 odd years now that when T cells are activated in mesenteric lymph nodes, they are programmed with a series of receptors that mean that those T cells will home back to the gut rather than to other tissues. And two of the receptors that do this are a chemokine receptor called CCR9, which recognizes a chemokine called CCL25, which is produced in the gut, and an integrin called alpha-4 beta-7, which binds to an endothelial adhesion molecule called mucosal addressing cell adhesion molecule 1, which is almost exclusively expressed in vessels within the gut. So this allows these T cells to go back to the gut, and that maximizes their chance of re-encountering the antigen which originally primed them. And these are long-lived cells which will recirculate for many years. And I could look in your blood now and isolate these cells from your peripheral blood and know that those are the cells in you that are providing immune surveillance within your gut. And that's in contrast to other populations of cells, and particularly cells that have been activated in lymph nodes draining the skin, which express a different complement of receptors which take those cells back to the skin. So there's this neat compartmentalization of immune responses and immune recirculation, which maximizes the chance of T cells encountering their triggering antigen again. So we wondered whether in liver disease complicating IBD, and particularly in PSC, whether this tissue-specific homing pathway might be dysregulated, resulting in these mucosal T cells entering the liver. And work done now several years ago by Alistair Grant and Bertus Eckstein, when they were in the lab with me, demonstrated that in PSC we do indeed see a change in the blood vessels in the liver. They begin to express these gut-homing molecules, MAdCAM1 and CCL25. So the liver now begins to look to the circulating mucosal T cell like the gut. Moreover, if we look within the livers of patients with primary sclerosing cholangitis, we can find populations of these mucosal activated T cells that are CCR9 high, alpha or beta-7 high, surrounding the bile ducts within patients with PSC. So this suggests that under the right circumstances, T cells activate in the gut to be recruited to the liver and drive inflammation. But of course, the next question is, what is it that induces the liver to express these gut-homing molecules? And here there was one of those remarkable coincidences in science that you could never plan in a grant application. So the molecule which we think is critical in doing this is a molecule called vascular adhesion protein 1 that I've been working on independently since 1996. And this is an endothelial adhesion molecule. It's constitutively expressed within the liver, where it drives the recruitment of a range of inflammatory cells, but particularly T cells, in liver diseases. And we've shown recently that this is a really important target in driving inflammation-driven fibrosis in liver disease. And indeed, there's a study underway by Boehringer Ingelheim based on our work looking at an inhibitor of this molecule in patients with NASH and steatohepatitis. So it's beginning to move into clinical trials. So this is an endothelial adhesion molecule that recruits T cells to the liver, but fascinatingly, it's also an enzyme. It's an ectoenzyme, it's an amine oxidase, and it sits on the endothelium that lines the blood vessels in the liver, and it has amine oxidase activity. And what it does is it takes primary amines and it generates aldehyde, ammonia, and hydrogen peroxide. And initially, we'd got no idea what this meant. We knew that some of the potential substrates for this enzyme were amines from food and wine and bacteria, which I'll come back to, but we really didn't understand what it meant. And Chris Weston, working in the lab with me for more than 10 years now, has really begun to dissect what this molecule is doing, and it comes back to the gut and liver axis. So Palak Trivedi, who's a clinical research fellow funded by the Wellcome Trust with us a few years ago, looked at the role of VAP1 in patients with primary sclerosing cholangitis and demonstrated that not only, as we've reported before, do you see this molecule in the normal liver, but it's markedly upregulated in patients with primary sclerosing cholangitis. And you can see that on the immunohistochemistry here, on vessels and on the fibrous scar tissue. But also the enzyme activity in the liver was markedly increased in PSC as well. So what is this enzyme doing on the endothelium? Well, when we began to look at liver endothelial cells that express the enzyme, and we began to trigger and activate the enzyme with a series of substrates, we found something very interesting. We found that the products of that enzyme activity, particularly hydrogen peroxide, signal back into the endothelium and activate a whole pathway of transcription factors, which results in an increased production of chemokines and adhesion molecules. And remarkably, one of the adhesion molecules which is upregulated is MAdCAM1. And we've been unable to find any other signal that would consistently upregulate MAdCAM1. So that's fine doing that in the lab in vitro with isolated endothelial cells, but what happens in vivo? And this is work that Leah Liascu published a few years ago now, where she took transgenic mice where we'd overexpressed this enzyme VAP1 on endothelium throughout the body, including in the liver, and then fed these mice with amine substrates for the VAP1 enzyme. And what she found was that MAdCAM started popping up in all sorts of places, in vascular beds where it's not normally expressed, including in the liver. So demonstrating in vivo that by providing substrates to this enzyme, we can change the pattern of adhesion molecules expressed by vascular beds, including the liver vascular bed. So we now have a model where we think that there may be an important signal, vascular adhesion protein 1 activation, which is driving this expression of MAdCAM1, leading to the recruitment of these mucosal T cells into liver tissue. But then the next question is, what is it that's activating VAP1? Where are the substrates coming from, and what are these amine substrates? And at the time we started studying VAP1, we were unaware of any physiological amines that were good VAP1 enzyme substrates. So Palak again did an exhaustive screen of a whole number, this just shows some of them, of amines which we thought might be substrates for VAP1. And the one that really stood out as being the most potent substrate was an amine called cysteamine, which I confess at the time I had never heard of, although I should have heard of it. And those of you who are interested in inflammatory bowel disease probably are aware of it. So when we took cysteamine and we put it onto our endothelial cells that expressed the VAP1 enzyme, again, we saw an upregulation of MAdCAM1 expression very nicely. And when we flowed mucosal T cells over these endothelial cells, they bound and migrated across the endothelium. So cysteamine was a potent activator of VAP1 and the most potent activator that we'd come across in the biological systems as well as in enzyme kinetics. So what is cysteamine? Well, cysteamine is generated by colonic epithelium by an enzyme called vannin-1. And if you overexpress vannin-1, you drive cysteamine production and colitis, which you can then treat by depleting cysteamine. So we've got a link with bowel inflammation. But I think more interestingly, cysteamine is also released by bacteria, particularly proteobacteria and firmicutes, some of the bugs that we see in the dysbiosis of PSC and indeed of inflammatory bowel disease, as we've already heard from Holm in the previous talk. So how does this relate to PSC? Well, this is some work done by Nabil Qureshi and Gideon Hirschfield in our group, looking at the microbiome in patients with PSC compared with ulcerative colitis and compared with healthy controls. And what stood out amid a mass of data is that in PSC IBD, we see this increase in proteobacteria and firmicutes, exactly the sort of bugs that can make cysteamine. And this work has been supported by work from other groups, particularly the Oslo group, who have found a similar pattern of dysbiosis in PSC. So we think that cysteamine may be released in the bowel as a consequence of the dysbiosis in PSC, and that this amine can then come through the portal vein into the liver, where it can activate VAP1 and all those downstream consequences which result in mucosal T cells being misdirected from the gut into the liver. So I mentioned at the beginning that we have no treatments for PSC apart from transplantation. So how can we take all this information and develop new therapies? Well, I think we've got some really exciting potential. So we can look at these homing pairs, CCR9 and CCR25, and alpha beta-7 and MAdCAM1. And of course, these molecules have been targeted therapeutically for the treatment of IBD. And particularly vedolizumab, which is an antibody against alpha beta-7, is now becoming a standard treatment for patients with IBD. So these drugs are out there, and we can begin to look at these in the context of PSC as well as in IBD. So at the moment, unfortunately, despite some of us trying for many years to get the major companies to carry out proper prospective trials of vedolizumab in PSC, those trials have not been funded by industry. But we do have some evidence from studies where people have gone back and looked at patients within IBD trials with concurrent PSC to see if the PSC improves on vedolizumab. I have to say the evidence is poor quality evidence. Some of the studies suggest an improvement, others don't. So I think the decision on vedolizumab as a treatment for PSC is very much out at the moment. But this must be an interesting area to pursue in proper controlled trials. So what about other therapeutic approaches that we may be able to deliver based on what I've told you? Well, one, of course, is to target VAP1. As I mentioned, Berger Ingelheim are looking at this molecule in steatohepatitis. And we are coming to the end of a multi-center phase 2 clinical trial looking at a fully humanized antibody against VAP1 in primary sclerosing cholangitis. And it will be very interesting to see whether we get a signal of efficacy in these patients. And finally, and this leads back to Holm's talk previously, the microbiome, as I've shown you, may be having a critical role in this process. So can we do anything to modulate these bacteria-derived amine substrates? Well, amines are incredibly difficult things to manipulate locally or within the circulation. But maybe one approach is actually to change the bacteria within the gut in patients with PSC. And this is a study actually in pediatric IBD, where the authors used fecal transplantation to treat children with IBD. And if you look at the panels here, you've got the outline of the microbiome in the donor, the recipient, and then sequentially after the fecal transplant in the recipient. And the red bars represent proteobacteria, potent producers of cysteamine. And you can see that a week after fecal transplant, the microbiome had shifted more towards the donor with a reduction of these proteobacteria. So maybe fecal transplantation is a way of modulating not only the microbiome and the regulatory networks which Holm described so eloquently, but also a way of disrupting this whole pathway that I've been talking about. So I want to finish there. We haven't come to a final conclusion as to how we can manipulate these pathways therapeutically, but I think there are some really exciting opportunities. And this has been a large body of work going back over 15 or so years now. I've mentioned some of the people who've done the work, the others are mentioned on this slide. And just at the bottom left, particularly to thank our patients who have painstakingly agreed to donate samples to us over the years, which has allowed us to do these sorts of experiments in human-based systems rather than relying on some of the more artificial murine models. And thank you all very much for listening.